So the final part I want to talk about is recombination in phages. So you can also have genetic exchanges between different phages that are not, closely related to each other. So these phages have evolved separately. These phages could have different phenotypes. And so, what exactly do I mean by a phenotype? So we talked a little bit before how phages can have different types of plaques. So this is kind of an example of different types of plaques. So this HR produces this very large, dense colored plaque. This H plus R has a very large but very light colored plaque. And then you kind of have the other different combinations as well. And so the reason for this is just that the phage has a genome. This genome can contain genetic variation, and this genetic variation can affect how the phage infects and acts. And these can lead to certain phenotypes. So, this phenotype is just in terms of the plaque shape and color. So, minute and turbid. So these are the two phenotypes that we just looked at above. But there's a large number of other phenotypes that have been observed and described in phages. So star is kind of the shape of the plaques. And then you can also have phages that are resistant to various degrees to things like UV, acroflavin, osmotic shock, lysosome, temperature sensitive, so what temperature they occur, and then even like different strains that they can infect. So this is a cool little mutant, so called the amber mutant, and this just determines which strains of E. coli it's able to infect. So these are both E. coli species. These are bacteria. K12 and B, they'll have genetic differences between them, and so they're different strains. They have different phenotypes, and somehow this leads to a difference in the ability of the phage to infect both of these E. coli cells. Researchers were able to demonstrate that genetic exchange can also occur in bacteriophages. And so, the only way that this is possible is for a cell to be simultaneously infected by two different viral particles. And so, you have the cell, this poor cell gets infected by a cell that is HR, and then unfortunately for it, it is also infected at the same time by a cell that is a different genotype and has a different phenotype. And so now you have two different genomes present in the same cell. So one has the H plus R plus, and the other has the HR alleles. And so now that is in the cell at the same time, now you have the opportunity to actually mix and match these genomes together. And so this is what I'm referring to when I say, genetic exchange between viruses. And so why is this important? Why should you care about this? Viruses are evolving all the time, and one of the things that they evolve is the ability to infect different hosts. So coronaviruses, for example, those are primarily found in bats and birds. So this virus is very, very effective at infecting bats and birds. And so typically, coronaviruses don't actually infect humans. And so, for that to happen, some sort of evolution must occur. And what evolutionary biologists think is important is that it's very difficult to kind of have a directed evolutionary route from infecting bats to infecting humans. But usually what you need to have is kind of like multiple genetic changes occur for this to happen. And so, if you read anything about kind of like some of the thoughts on how coronaviruses might have evolved, there's like a different organism that it might have jumped from. And so, why this is important is all of a sudden you have, and I think pangolins, I forget how, pangolins, is one possibility where this could have occurred. And so why would something like this be important? Well, you have, a virus, a coronavirus that infects pangolin. You have a coronavirus that infects that infects bats. And if there's some sort of, exchange that allows this bat to infect a pangolin cell at the same time as like the normal pangolin virus, now you all of a sudden have the chance to combine, evolution that occurred in pangolins and evolution that occurred in bats, and this could, potentially be something that creates something novel that can now infect a whole nother species entirely. I mean, typically this wouldn't be humans. Typically this would just be like, all of a sudden you evolve something that infects pigs or chickens or cows. And, this year, unfortunately, something new seems to have been evolved that is able to infect humans very effectively. And so how does this genetic exchange occur? So let's say you had two phages with the same amber phenotype. And so how did they demonstrate this? So you had a gene, the amber gene, and you had two independent mutations in it. So one of them was in this start of the gene, one of them was in the end of the gene. So just imagine these are two different viral particles. And so these co-infected a cell, and a single crossover occurred. And so now you're able to create two new combinations depending on where the crossover occurs. So these recombinants, in one case, you have a gene that has two mutations, but then the other recombinant would have a gene that has the wild type copy restored because you've lost both of those null mutations. And so as a result, you've rescued the phenotype. So this particular virus is no longer have the amber phenotype. It will grow on both the E. coli B and K12. This continues to have the same amber phenotype. And so this is where, having a large amount of infection where you have lots of viruses infecting an organism at the same time can be very dangerous because you're able to create kind of these new unique combinations of DNA that hadn't ever been seen before. So you're able to kind of mimic the benefits of sex even in viruses. The benefits of sex are thought to be like increased genetic exchange and increased genetic diversity. And so I think that is the extent of kind of what I wanted to cover. So this, I think, will be a very difficult chapter. So we're going to spend 2 full days working on the activities, and we'll have plenty of questions, plenty of time to